session for breaking down amino acids. So we're gonna have a great time.
Also known as transamination.
Yes, also known as
Urea
that.
cycle.
Yeah.
Yes, transamination terminating in the urea cycle. All right, so concept check. Why do we want to break down protein again?
For energy.
For energy, that's one reason.
Too big to store?
Too big, yeah, we don't have any place to store it. We're not going to build muscle, which takes energy to keep going, unless we need it. And so we're not going to just, oh, we have protein, let's make muscle. As great as that would be, it would actually be terrible. So those are the two reasons. We have too much, or we are really desperate for energy. So starvation or overabundance. Okay, so with that, as a really quick overview, we're going to start with pulling amino acids either from diet or muscle. Then they're going to be moved to the liver, and the liver is going to do some stuff to them. And then they're going to be moved to the kidney, and then the kidney is going to do the urea cycle to turn all the nitrogen, which is toxic if it's free, into a non-toxic substance called urea. And then that's the urea cycle, and we excrete it. So that is what we're trying to do. In the liver region is when you're going to be taking the energy out of the molecules. So getting some energy, getting rid of toxic substances. That's what we're doing. All right, so let's go ahead and start in the muscle. This is my muscle. And the muscle, made up with plenty of protein. And let's say that we are in starvation. And so we really need energy from somewhere. So the muscle is going to take a whole bunch of amino acids, which I'm just going to abbreviate A. And it's going to transfer nitrogen groups off of those amino acids. As you might remember from last time, an amino acid has an R group, it has a carboxyl, it has a nitrogen, NH2, and then it has a hydrogen. And so this nitrogen right here is what we are removing off of all these amino acids, and we're going to take that nitrogen and put it onto pyruvate. So if we add a nitrogen to pyruvate, pyruvate is actually this portion of an amino acid. So if you stick a nitrogen on the pyruvate, we will finish making an amino acid. Does anyone know what amino acid that is?
Glutamate. No.
Good guess.
Alanine.
Alanine, I heard it. Alanine. So when you think muscles, think alanine. Because the muscles are going to make a ton of alanine, and that's how they're going to ship their nitrogen. Okay, they're going to put a bunch of nitrogen onto pyruvate, makes it into alanine. And some of the other answers I heard are going to be right answers momentarily. So, pyruvate is going to pick up all the nitrogen, amino acids. If you take a nitrogen group off of an amino acid, what does it become? In general.
Oh, just the skeleton?
Carbon skeleton, also known as an alpha keto acid. Excellent. So I'm going to write alpha keto acid because that's what you'll see, but carbon skeleton is something that is often said. Alpha keto acid. Incidentally, it's called a keto acid because this right here becomes a double bond oxygen, and that's a ketone. So, pretty sure it's right there. There's a double bond oxygen near the nitrogen. I think it might be one more down, because that would be an aldehyde if it was on the end. Anyway, there's a ketone that's in there. All right, so alpha keto acids, examples of those are like succinyl-CoA, oxaloacetate, pyruvate, those sorts of things, where all of those can be used by the muscle to just make energy. So the muscle will go ahead and keep its carbon skeletons, but it's shipping off all the nitrogen to the liver, okay? And it's going to ship that by taking a whole bunch of pyruvate and changing it into alanine. And alanine is safe to go in the blood because it's, it's a protein. All right. So we're going to go ahead and bloodstream. Shift this off. Now, really quick, right here, we're going to have an enzyme that does these things. So box type objects are enzymes. So this one is just going to be various because you have a lot of different amino acids. So we're just going to say various transaminases. Okay. However, pyruvate to alanine, this is all doing the same thing. And so the very last transaminase, so there are pathways. So each amino acid can't necessarily directly do a transamination reaction with pyruvate. So there's occasionally a chain of action there. That's why I'm saying various, because they can do some trading. But once it is the last step for pyruvate to become alanine, that is going to be alanine transaminase, so ALT. And this is one that you guys will really want to remember more than just for the test. You're going to see this come up a lot.
Yeah.
So this is something that you can look for in the blood to say, hey, well, if muscles die, they're going to let out all the contents of their cells. ALT and also AST, which we're not talking about yet, but those two are found only in muscles and the liver, where we're going to head. So if you see those in the blood, then that means one of those two guys is dying.
So the alpha-keto acid is just the carbon skeleton of the amino that you put that in.
I got one.
Yes, so the alpha-keto acid is just the carbon skeleton of the amino acids, and then that'll just get chewed up for energy, so I'll just go ahead and, honestly, I think it makes it messy if you put this part in, but know the concept, chewed up, ATP. And then all the pyruvate that was there for other reasons, just because the muscle has pyruvate in it, you're converting a bunch of pyruvate into alanine. So, yeah.
And so the nitrogen group goes with glutamate.
Yes. So today is the game of follow the nitrogen, because that's literally all we're doing is it's like a relay race of, here, you get the nitrogen, now you get the nitrogen, now you, oh no, he's passing me, no. Chariots of fire, play, all right.
Hey Taylor,
So, yeah.
ALT stands for alanine transaminase.
Yes.
Okay.
And when you make that alpha keto acid, you said it chews up a lot of ATP there, or no?
So the alpha keto acid can be used to make ATP, because
Oh, yeah,
lots
'cause
of the alpha keto acids go into the TCA cycle.
Yeah, it's a ketone.
Yeah, it is a ketone, and then a lot of the molecules that they become are the ones in the TCA cycle. Occasionally it takes a few steps to get there, but if you turn one of them into oxaloacetate, then that's right there. So you
Yeah.
can just go through and
Yeah.
burn it.
There's
Yeah.
succinyl-CoA you said too.
Succinyl-CoA, yep, fumarate, pretty much all the TCA intermediates, some amino acid becomes them.
And you're saying that the keto acid, what about the chewed up ATP?
Chewed up equals ATP, so basically you just
okay.
use this for ATP, and I almost didn't write that in there because it's like, yeah,
Wait,
makes it a bit
so
messy, but it
you're
doesn't.
using ATP or you're,
You get ATP out of it because you are taking apart a carbon
Okay.
skeleton, and so all the bond energy, you're going to be getting energy from it. So
Could
that's
you
not the
say
super important part. The super important part that you actually really want to focus on is that the amino acids will give pyruvate their nitrogen, which makes pyruvate into alanine. Then we're shipping off alanine. That little thing right there, amino acid to pyruvate equals alanine, that's the big picture. And the enzyme that does the last step for all of it is going to be ALT, alanine transaminase.
You said something about AST, what is that?
We will get to where we see AST, but it's the other clinically relevant enzyme. So AST is going to be aspartate transaminase. And then this one is alanine. So the first two letters are like alanine, and then transaminase, and then aspartate, aspartate transaminase. So those ones, that's what we test for liver damage. We pull blood serum. Oh, they have elevated AST and ALT. They either have liver damage or muscle damage or both. And so that's a really powerful test, because when the muscle cells break, they dump everything, including their enzymes. All right, so any other questions? Okay, so let's, in the simple terms, go ahead and review the simple part. The rest of it you can kind of reason out, but just amino acids give nitrogen to pyruvate, and you get a ton of alanine. So that's what I want you to focus on here. And alanine is just a nitrogen carrier to go to the liver. So that would be part one. So go ahead and do active recall, and I'm going to draw a couple things while you do that. Things that look remotely like a liver. Knew I could draw pelican.
Or something. Does the alanine
Divers. All right.
Oh, that's a good question. Yeah, wait, so amino acid, isn't alanine an amino acid?
It is. So we're taking all the amino acids and we're making them into one particular amino acid.
Oh, because the amino acid is like a sequence, that other ones are like a sequence, or wait, no.
So
We're just removing the nitrogen.
I think the reason we make them all go into alanine is so that we can have a bunch of just a couple enzymes to make so that, okay, would you rather have, so I'll finish saying this and then I'll do the analogy, but so that you only have to have a couple enzymes in large quantities or regulated to say how much you want to do this process, rather than having 21 different amino acids regulated and having their enzymes in particular levels.
Mm-hmm.
So it's much easier to bottleneck it to one amino acid, because then you just regulate right there and the entire pathway is regulated. And yeah, so kind of similar to if you're trying to move, you want to put everything in boxes because it's much easier to move boxes and they fit very nicely, and this isn't the best analogy, but go for it. Whereas if you don't put anything in boxes, then you have 15 jillion things and nothing fits in the truck. I don't know. We won't repeat that one. All right, so alanine is going to go through the blood and it will arrive at the liver. And it's going to find a TCA intermediate, alpha ketoglutarate. Okay, and it's just going to give its nitrogen to alpha ketoglutarate. And it will do this using the enzyme should be glutamate dehydrogenase. So that's good. Oh, hang on, that's right. Okay, since it's doing a transaminase reaction, this is going to be ALT. And for those of you who just wrote in pen, my apologies. All right, alanine transaminase. So it's the same thing except now backwards, and you're moving it down to glutamate. So alanine is going to become pyruvate.
Yeah. So this is the Urea Cycle we're doing right now?
Exactly.
Ooh.
This is the Urea Cycle. Alanine from muscles to liver. That whole thing. Spoiler, Urea Cycle.
Sorry.
No, that's great. All right.
That's the alpha ketoglutarate right there?
Yes, that says alpha ketoglutarate, and if we add a nitrogen to it, that's going to become glutamate.
So this isn't just a transamination reaction?
It is a transamination reaction. So we are taking a nitrogen off of an amino acid and putting it onto a non-amino acid that is a carbon skeleton. And as soon as you add that nitrogen, alpha ketoglutarate, the only difference between it and glutamate is that alpha ketoglutarate looks, it doesn't have a nitrogen. So alpha ketoglutarate looks like this. No nitrogen. Glutamate has a nitrogen. So we are just changing alpha ketoglutarate into glutamine.
And so moving it to the liver is the Cori cycle part.
Yes, moving the alanine into the liver is the Cori cycle part, and the last part of the Cori cycle bit is alanine. I mean, all of this is Cori cycle, but the point when you're thinking Cori cycle, usually you're thinking, I'm making glucose, because now pyruvate can go and do gluconeogenesis. Awesome.
So the alanine transaminase, you're going to get two products from that. So you'll get pyruvate, which is from
Leftovers of alanine.
leftovers, and then
Gives it off.
glutamate from alpha-ketoglutarate.
Yep.
Okay.
So alanine just comes and bumps with alpha-ketoglutarate, and that shifts the nitrogen over, and then you just change the name of both molecules, even though they almost look the same as they did before, except the nitrogen is on the other guy.
Yeah, so when the nitrogen jumps onto the alpha-ketoglutarate, the alpha-ketoglutarate is now a glutamate.
Yes,
Yeah.
exactly. All right, and then pyruvate, we'll go and do pyruvate things with it. And this reaction right here takes, or let's see, that one produces an NADPH, and or NADH, excuse me, NADH. And I'm mentioning this, and I don't know that she will test you on this, but you remember when we were talking about the alcohol metabolism?
Mm-hmm.
How you shut down all the NADH pathways?
Mm-hmm.
This is one of them.
Mm.
And so this NADH being produced is going to be why the Cori cycle gets shut down, is because when you drink a bunch of alcohol, you have a whole bunch of NADH produced, and then you shut down any pathway that makes NADH. So just a bit of review.
Okay.
All right. Okay, so
So the alanine to pyruvate is ALT, not the alanine to alpha-ketoglutarate, or both are with the ALT?
really this whole thing is being done at once, because
Okay.
I might write it in the traditional way. I thought this would be better, but now that I got it up here, it looks kind of like a mess. But basically, this is making the nitrogen switch, and then as soon as the nitrogen switches, then this is called pyruvate, alanine is called pyruvate,
Oh, okay.
and alpha-ketoglutarate is called glutamate. So this enzyme is the one moving the nitrogen,
Okay.
and then everything just changes names, and that's why we have a bunch of names up there.
Is this so when you're moving forward to get, and you get NADH, you go the other way, you get NADPH?
You go the other way and you would spend NADH, so it would become NAD. So going this way, you get NADH.
That's oxidate, that's the dehydrogenase then.
There are multiple names for enzymes that can do this particular one. There's also, I believe it was pyruvate dehydrogenase. And actually,
Isn't that
this
the
isn't the
same-
same enzyme. So I know that pyruvate dehydrogenase does the last thing. Perhaps in her slide she skipped a bit there.
There was one,
Or-
I remember there was a slide where she was talking about oxidative reaction with the NAD and then, so, and I was kind of confused about it, but.
That's, what she was talking about was this,
Oh,
Okay.
glutamate
and you get
dehydrogenase.
NADPH plus hydrogen NADP, and then here you get NADH plus hydrogen NAD when you go the other way.
Okay.
That's what I was asking about, but it was the wrong way.
Is that,
Yeah, that
is
one's
that
without
this step or
ketoglutarate and
no?
glutamate.
Mm-hmm.
This one would be when you're deaminating
Okay,
rather than transaminating.
okay, okay,
And so right now we're transaminating,
okay.
which is this bit, and yeah.
So is the enzyme is either pyruvate dehydrogenase or ALT?
Yep, and I'm gonna have to double check on that because I've got two things that are conflicting.
Okay.
So I apologize for that, but hopefully the concept makes sense.
Oh, yeah.
And
That's detail. I would say.
if anyone can verify one way or the other, then that would be great. But we will continue on with glutamate here is going to have two things that can happen. Was there a question?
Yeah. So the alanine can change into pyruvate and alpha-ketoglutarate?
So the alpha-ketoglutarate was already in the liver,
Okay.
So the ones on the top are the ones that came from the muscle. The ones on the bottom are the ones that were already in the liver.
Okay.
It's just the nitrogen moving.
Oh, it's the nitrogen moving to the alpha glutamate.
Exactly.
Yeah, that's it. And then it becomes
from the
pyruvate.
alanine that was removed.
Yep.
Gotcha.
So whole thing is a relay race, and you have to write a ton of words, even though it's very simple, because as soon as you move a nitrogen, it changes all the names. So you have 2 names for before the nitrogen's moved, 2 names for after the nitrogen's moved, because as soon as you move the nitrogen, you change all the names.
So the alpha ketoglutamate, they were inside of the liver, and then the alanine gave the nitrogen to it?
Say that one more time.
The alpha ketoglutamate, it was inside of the liver?
Yes.
And then the alanine came and then gave the nitrogen?
Exactly. Yep. Alpha ketoglutarate was in the liver, and then the alanine came and gave it a nitrogen, which then pushed it to the other side.
To glutamate.
Yep. All right. So, good so far? All right.
This has been talking about, I tried to look it up, but it's talking about glutamase and glutamate dehydrogenase and for glutamine. And then here, it just says that transaminases for glutamate.
So,
But the one before that, it did talk about the alanine transaminase, so I think it's just alanine transaminase for the turning it to alpha ketoglutarate, giving it something, right?
alanine transaminase. Yeah. So, this part, this is
Carried out by ALT.
This is ALT. I think, I'm wondering, oh, okay. Okay, so all of this is done by ALT. You can also use pyruvate dehydrogenase if you don't want to put the nitrogen on the alpha ketoglutarate. If you just want the nitrogen to be free floating, you would use a different enzyme.
But you would never want that, right?
It depends. You don't often want nitrogen to be free, but it does have a pH balancing effect, and so the body could do that. But that's why we aren't talking about pyruvate dehydrogenase in this pathway, is because usually you're going to be, when you're doing this pathway, you're going to have a lot of nitrogen in the system, and so you don't want free nitrogen because you're already going to be making some along the way, which we're about to talk about. And so you're going to be using ALT because that's how you're going to get it to the urea cycle, by making glutamate. If you use pyruvate dehydrogenase, you would not make alpha ketoglutarate into glutamate. So how can I put this up here?
Did you say it's just it's starvation or fasting and
I
starvation?
will. This is going to be starvation, ALT,
Or overabundant.
or overabundance. So ALT is going to make the whole thing happen. Pyruvate dehydrogenase is just going to do the top half, and then it will release a free nitrogen.
Which is deamination.
Yes, which is a deamination. So it's a difference between a deamination reaction and a transamination reaction.
Gotcha.
Thank you for looking that up.
No problem.
All right. So, going to erase this ALT so that we just have it in the middle where it's like both. And then that one is just for this top. And that also explains why the NADH is made with the dehydrogenase. Okay, great. So, recap, make alanine, change alanine into glutamate. What the heck does glutamate do? Okay, glutamate goes to 2 places. Now, glutamate can become free nitrogen, NH4 ammonia, and this would be with the glutamate dehydrogenase. And so if the body is like, okay, we can regulate this one really well, if we want a little bit more ammonia to be released to keep our pH proper, then we can use this enzyme, and this is a highly regulated enzyme during the pathway, and so it's safe to have 20 to 40 millimoles, was it millimoles or nanomoles? Millimolar, I think it was millimolar. All right. This is what we usually have, is 20 to 40 millimolar, thank you. And then the toxic limit is going to be 400 millimolar. So if you go over 400, so 10 times higher than this, then you're going to start having toxic effects. And I'm going to go ahead and talk about those toxic effects after we finish glutamate getting its other branch done. Okay, so one is it can become free ammonia using glutamate dehydrogenase. By the way, this is 2, and now I consider all of this kind of moving on to part 3 if you wanted to just cut it out. All right, the other thing glutamate can do is there are 2 amino acids that can pick up not 1 nitrogen, but 2. And so glutamate is one of the ones that can pick up another nitrogen. And so glutamate will become glutamine. Excellent. And the enzyme is beautifully named Glutamine Synthetase.
And is this still happening in the liver?
Yes.
Okay.
So the liver is either letting out a little bit of free ammonia, or it's packaging the nitrogen even more condensed. So alanine can only carry 1. The liver's like, okay, let's jam as much as we can onto 1 molecule before we send it on to the kidney, which we will talk about. So this makes glutamine, which will go into the blood and go to the kidney. Any questions? Okay, cool.
Can you say what the millimolar is again?
So this is normal. This is toxic limit. So if you're a 20 to 40 millimolar, that's normal to find in the blood. 0.
That's ammonia.
Huh?
Ammonia, right?
Yeah. And if you have 400 or more, then you got some problems. And we'll talk about those problems right now, I think. So, ammonia, that is going to be basic or acidic?
Basic.
Basic in this case. So, ammonia is, so if you look at, where is there? So see how it starts as an NH2? And then it's popped off? How did it become NH4?
More hydrogens. Free nitrogen.
More hydrogens. It picked up free hydrogens, and so you have fewer hydrogens in solution.
Oh, yeah.
This is more than she's going to ask. I just like background stuff.
Yeah.
So if it picks up hydrogens, then that means it's taking acid out of solution. Therefore, it's a base.
Yep.
So the solution becomes more basic because it's grabbed all the acid out of it, because hydrogen is what acid is, basically. So if you ever are like, is this an acid or a base? It's grabbed hydrogens out of solution, therefore it's a base. Okay? For, yeah. So, or had a basic effect as it was created, might be more correct. So this was the base and then it's already grabbed it all. Now, so the more nitrogen you release, the more basic your pH. So that's one thing that it'll do. Issues. Number 1, pH goes up. Number 2, so nitrogen, if you added nitrogen to alpha ketoglutarate, you would get glutamate, which we've talked about. And it turns out that in the brain, this enzyme is not regulated enough to stop this happening if you have a freaking ton of ammonia. And so ammonia will go through the blood-brain barrier and into the brain. That already sounds scary. So let's just stop there and go to sleep and sleep well, but no. So,
Clean with Windex.
Windex, okay. Not making that connection quite yet, but it'll come. So, the, basically all the alpha ketoglutarate, which I'm going to do alpha Kg, in the brain will become glutamate. So in the TCA cycle in the brain, alpha ketoglutarate is a part of that TCA cycle. If you took all the alpha ketoglutarate out of the cycle, what would happen to the cycle?
Broken.
Broken. You get nothing.
No
And so the
energy.
brain is now starving for energy. And that's bad enough. But all the glutamate, what can glutamate become if you add another nitrogen? Glutamine. So it doesn't stop there. The glutamate, which you at least could use as a neurotransmitter, is now being changed into glutamine by adding another nitrogen. And I'm just going to go ahead and emphasize the nitrogen. So this is just all because nitrogen is forcing these reactions.
This is all in the brain right now?
Yes. So all your alpha-ketoglutarate turns into glutamate. All your glutamate turns into glutamine. And glutamine is not going to give you energy. It's not going to give you, neurotransmitter ability. But it will give you edema. Yay!
Oh, that's fine.
So
Want that.
I want edema, man. All right. So we're going to have H2O running towards the glutamine. And that's going to cause edema in the brain, or swelling of the brain. And that can get so bad that your brain actually herniates through the foramen magnum. And then that's bad. So those are the three issues with having lots of free ammonia. Okay? So, any questions on those? All right. Oh, question?
Yeah, I'm a little bit confused. So the glutamate that we had from the alpha-ketoglutarate gas, does it do this or that, or is it doing both of those things?
This or that. And you are not typically, so the glutamate isn't going to cause this to go to toxic limits. It's going to go to safe limits.
Okay.
Come to think of it, yeah, we'll come to the part where it can get to toxic limits. I just got excited because I think it's a cool story that I hope never happens to anyone in this room. So, yeah. Any other questions?
And then how is that kind of like regulated? Not that I want to get into something that we're not learning, but so how does it know when to go to glutamine?
So I've never read anything specific. I would guess that it's negative feedback. That's how most of these things are regulated. So, we have
Enough?
40 millimole of ammonia. Let's turn this off.
Okay.
That's going to be a negative feedback loop of some kind.
Okay. Thank you.
All right, so
That's glutamine
synthetase. And so if it's synthetase, does it take ATP? Synthetase, yes. That's just a little fun detail. All right, so going to the kidney. In the kidney, glutamine arrives.
So step 2 happens in the liver, and step 3 in the brain. Step 1 in the muscle, 2 in the liver, and
So I was thinking 1 was muscle, 2 was liver, and then 3 was, let's have 3 in the brain.
Well, 3 is still in the,
This part is all liver.
yeah.
The issues in the brain are
Oh, it's, oh.
with just the ammonia. So, yeah.
so that part is the issues in the brain.
Yeah, we'll go ahead and say 3 is in the brain because I think at this point that makes more sense. And so really the way that you break it up is whatever makes sense in your head. So I'll just do this and then it's clear. I didn't have time to break it up properly. So,
The glutamine, number 3, so it attracts water?
yes. It's just the nature of the molecule. You build up enough glutamine, glutamine is going to be present and that attracts water because, I would guess, because it has polar groups and water is partially positive. It has partial charges and so it's attracted as glutamine.
But it's not the same case with kidney.
Kidney's okay with extra stuff because, number 1, it's going to break the glutamine down pretty fast, which we're getting to. And number 2, the kidney's kind of where you excrete water out of the body, so it's good at handling water.
Okay.
All right, so glutamine. As soon as glutamine arrives, we are going to use glutamine, glutaminase, glutamase. So glutamine ACE. Okay, so we're breaking glutamine down to glutamate, which means we released a nitrogen. And then we take glutamate and break it down to alpha ketoglutarate. And that's going to be done with glutamate dehydrogenase. And I'd like to take this moment to say again, the dehydrogenase, that's a deaminase because it's not transferring the ammonia, the nitrogen to anything else. So it's a deamination whenever you have a dehydrogenase doing the action, because that's just popping the ammonia off and it's not putting that on anything else. Whereas back here with the ALT, these are transaminations because you're moving the nitrogen from one carbon skeleton to another one. Does that make sense?
Wait, so dehydrogenase is deamination?
It is, in this case. That's one of the weird things about dehydrogenases. I've been wanting to look at exactly how it does it chemically. I imagine that it probably is either adding a hydrogen, which bumps the nitrogen off as a result, so it's still dealing with hydrogen, but I haven't looked at it.
Yeah,
Taylor, is the second enzyme, is it glutamine dehydrogenase or glutamate dehydrogenase?
Glutamate.
Glutamate.
And then these are just nitrogens coming out. All right. Alpha ketoglutarate is going to do its thing. It'll go in the TCA cycle, and we don't care about it. It's great. Now, however, we have free nitrogen, ammonia, floating in the kidney. Oh my. So the kidney is going to take that, and it's going to do the urea cycle. And before we go there, I think go ahead and see if you can walk through muscle, just the basic story. Liver, basic story. And then the third part, I'll cue you along. So go ahead and close your eyes. What happens in the muscle? All right, so in the muscle, we're taking all the amino acids and we're changing them into alanine. Then we're going to send alanine into the blood. Part 2, what do we do in the liver once alanine arrives? Okay, so alanine is going to give its nitrogen away to alpha ketoglutarate. And that means that we have to rename alanine pyruvate and rename alpha ketoglutarate glutamate. All right. And what are the two options that glutamate has? So the two options are glutamate can either release ammonia just out to be free, or it can pick up another nitrogen and become glutamine. All right. And while we're thinking about the free ammonia, what are safe levels? What are toxic levels? And then 40 times 10 is 400. That's how I like to remember that, just add a 0. Okay. And then the other option that glutamate could do was also glutamine synthetase, which is going to make glutamine, and then we'll continue from there in just a bit. But if I want to pause just for a memory thing here. So this is a decent amount of stuff so far, and so I find it helpful that if I feel like I'm going through a memory thing and it's like 1, 2, 3, 4, 5, and it gets to the point where it's like my head's starting to hurt and it's like, this is too big, I'm losing place of where I am, just like cut off the entire thing and call that, you know, just, it's kind of like chunking the chunks, and don't make it all part of one sequence, one after the other, to the point that it's too much all in one string. If it feels comfortable, keep going, but if it doesn't, then just go ahead and say, all right, what were the first several things? Okay, I have now accomplished the first several things, the first all the way to here, and I would call that a single item of a single review story. Does that make sense? And then the next one, I have a second review story, and it sounds like it's really pedantic, but it makes a really big difference in how much working memory you're using up with your previous parts of the story if you literally break it into, okay, one big chunk with a bunch of little ones, and then a second big-ish chunk with some little ones, and you just completely dump all the first things out of your head. Makes a huge difference. Sounds like it's doing the exact same thing, but it makes a big difference. So
Yeah. I know I do that when I'm studying. I get to a point and I'll just have to be like, okay, that's all done. I'm gonna start over, start fresh, you know, and here I go.
exactly.
I don't know, there's something about it that's weird.
All right, so continuing on with our second big thing, we're going to have brain and kidney. So, go ahead and active recall the brain, and you can do it in connection with the previous thing or isolate it or however you want to do it, but I've had my spiel. Go ahead. What is the problem that happens in the brain when you have too much ammonia?
Wait, so it increases the pH instead of decreases the pH?
Right,
That's
it's
because of
alkaline. Because the nitrogen grabs a whole bunch of hydrogens, and that's for the whole body, not just the brain, the pH one.
Oh, right. Yeah, increases, never mind. I knew that.
I think everyone does that. I still do that. I was like, oh, I know this answer really well. That was not very long ago, and it's like, dang it. Increasing acid, I don't know, don't ask direction questions. All right. All right. Second is it's gonna eat up all the alpha-ketoglutarate, glutarate, turn it into glutamate. Next, glutamate turns into glutamine, so it's just like 1, 2, 3, grows it into glutamine, and therefore you have no energy, you have no neurotransmitters, and then you have a whole bunch of edema from the glutamine. All right.
I have a question.
Please.
So the glutamate, when it goes into two different places, either the brain or kidney, would it go to the brain first and then the kidney?
So the glutamate is going to become either ammonia or glutamine. The ammonia just goes into the blood, and that means it will eventually go to the brain just through the blood, and the brain is where it has an issue. The rest of your body can handle it to some degree, or at least it's not as bad as it ends up being in the brain. If you die, it's going to be brain edema. Other tissues, if they have edema, it's not good, but they're not going to kill you. Glutamate to glutamine, that also goes through the blood, and the kidney just scoops it out. No one else tries to take glutamine out of the blood.
Okay.
Well, that we care about.
So glutamine goes to the kidney.
Yep. So this is back to the relay race thing.
Yeah.
Shredding off all the nitrogens and throwing them into the kidney solution, so now we just have a bunch of nitrogen in the kidney.
So what's the second? Yeah, glutamine what?
So you have glutaminase or glutaminase, and then you have glutamate, because now it's acting on glutamate,
Oh,
dehydrogenase.
the glutaminase is the enzyme that removes the nitrogen?
For the first one, because the first enzyme acts on glutamine, so we're going to name it after glutamine. Glutaminase, glutaminase. And then once it's glutamate, we're going to name the enzyme after what it's acting on. So glutamate dehydrogenase.
Okay.
and then it makes it into alpha ketoglutarate, and alpha ketoglutarate goes and does its thing. Question?
So the urea cycle, does it start from alanine to the kidney? Or?
Alanine.
The urea cycle is specific to the kidney. We haven't started it yet except this ammonia, which is an N at present, but adds some hydrogens to it. That would be the technical beginning of the urea cycle. So all of this is amino acid metabolism. The urea cycle itself is just take nitrogen, turn it into urea, which is what we're going to sort of get to in our remaining time.
Let's do it.
All right. So you actually have a really good picture in your slides for this one. That's the picture that I still go back to if I need to go back to something. And so I recommend pulling that up and kind of splitting it to mitochondria versus outside the mitochondria. And really quick, I could draw it up here, but basically I'm going to draw the same picture. So you have free nitrogen being combined with carbon dioxide and water. And then you're going to use an enzyme, and there are enzymes that do that. And those enzymes are important, so look those up. I ran out of time this morning. So with those two enzymes, those are the enzymes that if they're broken, you're going to have the edema of the brain. Because if you can't make the ammonia into urine, because those two enzymes are broken that are supposed to grab the nitrogen and make it into a, the first enzyme is carbonyl phosphate synthetase, and that one, yeah, you can't make it into a carbon structure that becomes urea. So if these two enzymes are broken, then you're just going to let that ammonia float out into the blood. You delivered it to the kidney can't do anything.
CPS 1 and 2.
CPS 1 and 2, thank you. CPS,
Is this the picture you're talking about?
wait. Carbonyl phosphate synthetase 1 and 2, hang on. That is the picture I'm talking about.
I think there's
I
only
don't
one.
think it's carbonyl phosphate synthetase 1 and 2
Oh,
because
there's
that's actually
CPS 1.
in,
The second one's different.
Yeah, it's right here.
There's one, but not two.
There's CPS 1, and then this one's orth.
right, but what makes the, before the carbonyl phosphate synthetase, you see right there,
Mm-hmm.
how it makes bicarb?
Oh, yeah.
What's the enzyme that makes the bicarb? And there's actually two, even though it's not showing on there.
Is there a CPS 2 or did I make
There
that
is
up?
a CPS2, and we will see that later. It's like, it says 1, there has to be 2 somewhere. And there is, and it's in a completely different pathway. It's like, okay, I gotcha. Okay, so the enzyme that makes bicarb, carbonyl phosphate.
That's all that she has. Like,
This one was always the hardest to find.
do we need it? Do we need it? If she, like, she didn't list it.
It is important, and maybe carbonyl phosphate synthetase 1 has 2 activities or something like that.
Is it arginine?
Okay, this C, and then it's still taking the bike. Oh, okay. CPS 1 is definitely one of them, so good. All right, because there's, all right. So carbonyl phosphate synthetase, if that's broken, then you can't make the first molecule, which is carbonyl phosphate. And yeah, so that would mean that the nitrogen is going to go free and stay in the blood because you can't take it and make it into a molecule. Does that make sense?
No.
All right. Let me
The nitrogen.
try and, yeah, I need more space. Okay.
You can have this.
All right, so we have gotten NH4, and then we also have bicarb. And putting those two together makes a molecule called carbamoyl phosphate. And the enzyme that does this is carbamoyl phosphate synthetase 1. If this enzyme is broken, then this nitrogen, this ammonia, is going to stay free. And that's what will end up causing edema in the brain. So this is a genetic disease. And yeah, then the rest of this is just going to be the urea cycle, where you take the carbonoyl phosphate and you have an ornithine molecule, which is, I think of someone who's ornery. I'm ornery! And I'm also, I don't know if that's Southern or who pronounces it ornery, but someone pronounces ornery. West?
Maybe Missouri.
Missouri? Okay, we'll say Missouri. That sounds good.
Okay.
I'll take it.
I don't know.
Citrulline. All right, so we take ornithine and we combine it with the carbonoyl phosphate. And that makes citrulline. And so that's what's going on there.
Okay. Does that get released in the E as well?
Sorry?
Does it get released in urine as well?
Citrulline is eventually going to become urea. So all of this is now urea cycle. And this part right here where you take the ornithine and put it together with the carbonoyl phosphate, that's the part that some people quirk their eyebrow at, so I wanted to hit that. But I do have to leave before hitting the rest of this. Basically, the citrulline will, all of this happens in the mitochondria of the kidney. So this is kidney. This is mitochondria. And then the citrulline, or citrate, or whatever, it says citrulline, citrulline. Okay, citrulline can leave the mitochondria, and so it gets outside and is going to be joined by aspartate. And so we put citrulline and aspartate together, and that's going to make arginosuccinate. Arginosuccinate will eventually become arginine. And arginine will have, it'll be acted on by arginase, which is nice, arginase. And I'm kind of going fast because I know you have this right in front of you. Urea. And then the arginine, this is the reason I'm doing it, is so that I can point out this. The arginine, not all of it turns into, the entire arginine molecule isn't poof, the entire thing's urea. You take a bit of it, and that becomes urea. The arginine then is going to come, and once you've cut off that bit, so to speak, it becomes ornithine. Ornithine. And you put the ornithine back in the mitochondria, and so it's this loop. So ornithine, you stick something on the side of ornithine essentially. Ornithine goes through all these other things, and you call it a bunch of stuff, but it's still got that thing on the side, and eventually you call it arginine, then you cut that thing off, and it's urea. And then you have so ornithine is just kind of carrying the nitrogens that carbonoyl phosphate is made of. And so ornithine picks up the nitrogens, goes over here, dumps the nitrogens, it's called ornithine again. And that's what this cycle is doing.
What's that one? The side behind?
That?
Yeah.
I was starting to write out the name of the enzyme, and then I realized I didn't have time. So ornithine carbonoyl phosphate transferase or something like that.
Did you
So,
get what that?
any concept questions?
Cooper, what is that one? Arginine
This is arginosuccinate, and I wrote super messy because it's in your slide, really, really beautiful, and it looks
Okay.
exactly like this. All right.
Thanks, Taylor.
Yeah, anytime. Good deal. I will open up the quiz in like a little bit. And I would recommend two things. Number one, getting your head around the aminotransferases. How an aminotransferase is just moving a nitrogen, and it changes, alpha-ketoglutarate into glutamate, alanine into pyruvate, and as soon as that is something that's comfortable, this entire thing becomes really simple. until that makes sense, it makes it pretty hard to get your head around. And that's what I found when I was going through it the first time. That was the one thing that tanked me and everyone I talked to.